everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is uptake and transport of mineral nutrients in plants. This video is taken from Plant Physiology series, CBSE class 11th, part 9th of chapter 12. Now, let's see here the lecture overview. So, in this video, we will focus on uptake of mineral ions, translocation of mineral ions, translocation of organic nutrients, evidences to support that translocation occurs through phloem, and the last is mechanism of phloem translocation, in which Munch's mass flow or pressure flow hypothesis. Now, let's start the topic, in which our first topic is uptake of mineral ions. So, the minerals are mainly absorbed by the roots, which are in the direct contact with the soil solution. Unlike the absorption of water, the minerals are absorbed over the entire surface of young root by the cells of epiblema. During absorption, the ions or molecules penetrate into the inner space of the living cells or tissues from the surrounding medium through a selective membrane. The movement of dissolved substances into and out of the cell is also called transport or flux. Various theories have been put forward from time to time to explain the mechanism of solute absorption. They are broadly categorized into two groups based on the involvement of metabolic energy in the process. Those which believe that metabolic energy is not involved in the solute absorption have been grouped under passive mechanism, and those who believe that metabolism and metabolic energy is involved in the process have been placed under active mechanism. So, the recent findings indicate that minerals cannot be passively absorbed by the roots because of the following two reasons, in which the first one is minerals are present in the soil mostly in the form of charged particles which fail to move across cell membranes. And the second is concentration of minerals in the cell is usually greater than that present in the soil solution. Therefore, most of the minerals are absorbed by active mechanism involving expenditure of energy in the form of ATP. Now, translocation of mineral ions. So, how the minerals ions translocate? At first, see here that xylem and phloem combine to form the vascular system. So, in case of transport, xylem tissue transports water and minerals. The mineral nutrients are taken up by the roots and transported upward into the stem, leaves, and the growing regions. While in case of translocation, this is the process of transporting food substances downwards from the leaves to all other parts of the plant through the phloem. So, transport by xylem and translocation by phloem. The absorbed minerals elements move laterally from epiblema to xylem through cortex, endodermis, and pericycle. Finally, they are transported upward to the xylem along with the transpiration stream. This has been demonstrated by the ringing experiment that upward transport of minerals continues even when the phloem tissues were removed. Analysis of the xylem sap also shows the presence of large amount of dissolved salts, and this includes some organic compounds of the nitrogen and phosphorus which are largely responsible for the movement of nitrogen and phosphorus. Some of the minerals are transferred radially from the xylem to the phloem mainly through parenchymatous cells, and they are also transported to other tissues where they accumulate in the living cells. Several evidences suggested that the minerals are transported through phloem also. The translocation in phloem mainly occurs from the leaves just before the abscission. So, if we talk about that generally believe that the mineral nutrients absorbed by the roots finally reach to the xylem after passing through the cortex, endodermis and pericycle. And because of this, there are two mechanisms of the movement of minerals from the soil into the dead xylem cells, in which the first one is apoplast pathway and the second is symplast pathway. The apoplastic pathway involves movement of minerals by the simple diffusion from cell to cell through their primary cell walls, which is made up of polysaccharides. Minerals move through apoplast in cortex and pericycle but not in the endodermis. The cell wall of the endodermal cells possesses a conspicuous waxy thickening which is known as the Casparian strips, which blocks the passage of solutes from one side of the endodermis to the other via this cell wall route. Therefore, minerals can enter the pericycle and the xylem by passing through the protoplast of the endodermal cells. Thus, the movement of solutes is apoplastic in cortex and pericycle but symplastic in endodermis. Now, the second one, that is the symplastic pathway, which involves the movement of minerals absorbed actively through the plasma membrane of cells of the outer parts of the root, that is epidermis, through the protoplast of the cortex, endodermis and pericycle, and finally released into the xylem. The cell-to-cell movement of ions in cortex takes place through the plasmodesmata. The term symplast refers to a system of cells whose protoplasts are interconnected. Now, after this, how translocation of organic nutrients occur in plants. So, see here the translocation of organic nutrients or solutes. The synthesis of carbohydrate food materials, mainly through the process of photosynthesis, occurs in the green cells of the plant. The non-green cells are therefore dependent on photosynthetic cells for their carbohydrate supply. The organic carbon, mainly from the leaves, is transported to the non-green parts where it is needed for the respiration and biosynthesis. The transport of organic solutes from one place to another in higher plants is referred to as translocation of organic solutes. It has now been well established that the carbohydrates are translocated from leaves to root and storage organs along the phloem in the form of sucrose. They are transported through living sieve elements of the phloem, chiefly sieve tube members in the seed plants. The process of translocation requires expenditure of the metabolic energy and the solute moves at the rate of 100 cm per hour. So here you can see that the movement from the sink to from the source to the sink. So, what is phloem loading? Where do the solutes come from? In a process called sieve elements, loading sugars are transported into the sieve elements and the companion cells. Sugars become more concentrated in the sieve elements and the companion cells than in the mesophyll cells. Once in the sieve elements or companion cells, complex sugars are transported away from the source tissue called export. Translocation to the sink tissue is called long distance transport. So how the growing parts of the plant are provided with the sugar to synthesize new cells? A system of vascular tissues run through all the higher plants. It involves as a response to the increase in the size of plants which causes and progressing separation of roots and leaves in space. The phloem in the tissue that translocates assimilates from mature leaves to growing or storage organs and roots. Now, evidences to support that translocation occurs through phloem. So, see here are some evidences which support that the organic solutes are translocated through phloem, in which the first one, which is known as the ringing or girdling experiment, which you can see in this picture. So the experiments in which the continuity of the vascular tissues is disturbed by their removal are known as ringing or girdling experiment. So see here, girdling usually employed from the removal of all the tissues outer to the vascular cambium, that means bark, cortex and the phloem, from the OD system. In OD system, that means stems with the single ring of collateral vascular bundles, it is easily possible to remove a broad band of the bark including phloem and the cambium. Under such conditions, the upper part of the plant is attached to the lower part by only the central xylem cylinder and pith. It results in the accumulation of food materials just above the ring, which is evident by the swelling and sometimes formation of adventitious roots. So in this case, furthermore, it can be observed that the roots die first in the girdled plant. This can be explained because the transport of water and minerals occur through xylem. The upper part get ample amount of water supply, the supply of minerals and all the important factors for the metabolism. It is therefore the upper part continues to grow. The girdling results removal of phloem, and since the phloem is supposed to translocate organic food materials, it cuts the food supply to the roots. The roots cannot synthesize their own carbohydrate and thus they die first. So, in ringing experiment, why is there swelling above the cut region? So, because of this, the removing of the bark would remove phloem. Accumulation of food substances would cause swelling above the cut region. Now, what are the result of the ringing experiment? The removal of the phloem prevents the transport in the plant. Now, the second evidence is the isotopic studies. If a leaf of a potted plant is illuminated in the presence of radioactive carbon dioxide, that means 14CO2, it forms radioactive products of the photosynthesis. These products with radioactivity are then transported to stem. It was detected by the autoradiography that these substances are translocated through the phloem and particularly through the sieve tubes. Now, the next is chemical analysis of the phloem sap. Chemical analysis of the sieve tube sap prove to be a sweet concentrated solution of sugar. This analysis support the evidence from the ringing experiments in pointing to the phloem as the main channel for the translocation of organic solute. So these are the evidences to transport that translocations occur through phloem. Now, after this, what is the mechanism of the phloem translocation? So, several theories have been proposed to explain the mechanism of the phloem translocation, like diffusion hypothesis, active diffusion theory, electro-osmotic theory, interfacial flow hypothesis, Munch hypothesis, etc. Among them, the Munch mass flow hypothesis is the most widely accepted, which is briefly discussed here. So, here you can see the mass flow hypothesis, which is described by Munch. The mass flow or the pressure flow theory was the first proposed by Munch in 1930 and later elaborated by Crafts in 1938 and others. The Munch hypothesis postulates the movement of protoplasm and mass along a turgor pressure gradient induced by a maintained gradient of the water potential. The mass flow of the organic solutes takes place from the site of higher concentration to the site of lower concentration. So, here you can see that a model which demonstrates the Munch mass flow hypothesis. So, in this model, the principle of mass flow hypothesis is illustrated here. That means the two chambers, that means A and B. Here you can see in this picture, with semi-permeable membranes are connected by a tube. These chambers are immersed in two reservoirs connected through a tube T. So here you can see that connected by the tube T. The reservoirs are filled with water. The chambers A contains concentrated sugar solution and the chamber B contains dilute sugar solution. The osmotic pressure of chamber A is high as compared to B. The water enters into the chamber A and its turgor pressure is increased. This increase in turgor pressure causes mass flow of the sugar solution to the chamber B under the influence of turgor pressure gradient. The movement of solute will continue till the solution in both the chambers attain the same concentration. So in the living plants, the carbohydrates are continuously synthesized in the mesophyll cells of leaves. Consequently, the osmotic pressure of these cells is increased, which then absorb water from the neighboring cells. The turgor pressure of the mesophyll cells is also increased. This allows some of the cell content to pass into the sieve tubes. On the other hand, in those parts like roots and storage organs, cells either consume food material or they convert it into insoluble storage forms. This transformation results decrease in osmotic pressure. With the result, their turgor pressure is also decreased. In this way, a turgor pressure gradient is formed between the supply site and the consumption site, and along with this gradient, a mass flow of solute takes place. So, in case of the pressure flow model, here you can see that a diagrammatic representation of the mechanism of the solute translocation according to the Munch hypothesis. The translocation pathway has cross walls, allow water to move from xylem to the phloem and back again. If absent, pressure difference from the source to sink would quickly equilibrate. Water is moving in the phloem by the bulk flow. No membranes are crossed from one sieve tube to another. Solutes are moving at the same rate as the water. Water movement is driven by the pressure gradient and not water potential gradient. So what are the steps in the Munch mass flow hypothesis? First one, that is the water absorption from the nearby cells increasing hydrostatic pressure in the mesophyll cells. Then phloem loading, transfer of the organic food from the mesophyll cells to the sieve tube. Then the phloem uploading, unloading, transfer of organic food from the sieve tube to sink. Then at first, after the phloem loading, the third step is water entry from the xylem to sieve elements by the osmosis. Then the fourth step is hydrostatic pressure differences drive solute movement from the sieve tube to sink. Then the fifth step, which is the phloem unloading, transfer of organic food from the sieve tube to sink. And the last step is water moves back to the xylem vessels by osmosis and transpiration pulls up water. So this is the long distance transport of mineral nutrients in case of plants. Now, after this, see here the Munch hypothesis, which has been supported further by the followings. When a woody or herbaceous plant is girdled, the sap containing high sugar content exudates from the cuttings. Positive concentration gradient disappears when the plants are defoliated. And the last is the movements of viruses and the growth hormones is fast in illuminated leaves as compared to shaded leaves. So these are the supported by the following some views. And after this, the objections to the Munch hypothesis. So what are the objections to the Munch hypothesis? First one, that is the hypothesis fails to explain bidirectional movement of metabolites, which is common in case of plants. Osmotic pressure of the mesophyll cells and that of root hair do not confirm the requirements. The last is Munch's hypothesis gives a passive role to the sieve tube elements and the protoplasm. So this is all about the uptake and transport of mineral nutrients in plants. This is the end of video in which we understood about the uptake and transport of mineral nutrients in case of plant and especially Munch's flow hypothesis. Now, the next upcoming video will be on the photosynthesis in higher plants. So from the next video, we will start photosynthesis. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.